sweeping the nation, the lack of transplantable organs. Every single day, 24 people die waiting for organs on the organ list. The current organ transplant list takes only organs from human donors. This means that organs are very scarce, and there aren't many of them. We had our own Wyatt Chang go interview a scientist who thinks he has a technology that can revolutionize the medical world.
So what are you working on over at the university?
What we've been doing here is a bit weird. Basically, we've been taking the blastocyst of pigs, which is pigs when there's still a couple of cells. These cells are embryonic and later can turn into any cell that the pig needs. Then what we do is we take adult stem cells from the patient and we insert them into the blastocyst.
So what does this do?
Transplanting human adult stem cells from the patient would allow the pig to develop human organs instead of their own normal pig organs. These we can then surgically remove from the pig and insert into the patient, replacing their own damaged ones. Now, instead of having to wait on an organ transplant list, scientists can engineer their own organs. And furthermore, because we're using their own stem cells, there is a much lower risk of organ rejection.
This form of genetic engineering is referred to as a pig chimera. It's come a long way since its discovery in 2017, and our friends at Scishow have made a video to explain it.
In one paper published on Wednesday in the journal Nature, a group of biologists announced that they genetically engineered a rat to grow a mouse's pancreas, then successfully used cells from that pancreas to suppress diabetes in mice. And in another paper published on Thursday in the journal Cell, a different group announced that they genetically engineered a pig embryo to grow with some human cells. There's a long way to go before we're able to grow functioning human organs inside of other animals, but both of these studies mean that we're a lot closer. Right now, the organs used in transplants have to come from human donors, but the donor pool isn't very big, especially for essential organs like hearts and lungs that can only be donated when someone dies. That's why lots of researchers are looking into ways to grow human organs inside of other animals so doctors could grow whatever healthy functional organs they need. The same team that published the Nature paper this week actually created the first combined rat mice back in 2010. These were the first rat-mouse chimeras to be grown from embryos and survive. To do this, they injected rat stem cells into mouse embryos that had been genetically engineered so none of the mouse stem cells could develop into pancreatic tissue. Those embryos grew into rat-mouse chimeras with mixtures of rat and mouse cells throughout their bodies except for their pancreases, because only the rat stem cells could grow into pancreatic tissue. When it comes to organs, the pancreas is relatively simple, so this isn't quite the same as growing and transplanting, say, an entire heart or lung. That would be much more complicated. But thanks to this research, we know that organ transplants from chimeras can actually work. That's just mice and rats, though. The goal of this line of research is to eventually grow human organs inside a species with similar-sized organs, like a pig or cow. And that's where the paper published in Cell comes in. This group of researchers wanted to create human pig chimeras by injecting human stem cells into pig embryos, but that's a lot harder than creating a rat-mouse chimera. For one thing, humans and pigs have much less DNA in common than rats and mice. For another, pig fetuses only take about 16 weeks to develop, while human fetuses take 40 weeks. So human stem cells and pig stem cells develop at different rates. All this makes it harder for human cells to survive and become a healthy integrated part of a pig embryo. For this study, the researchers injected human stem cells into pig embryos, then implanted those embryos into female pigs. They let the fetuses develop for just 3 to 4 weeks, then euthanized them so they could analyze their cells in the lab. They found that the chimeras had developed with some human cells, but not very many of them, and even fewer of them grew to a typical healthy size. The fetuses were mostly pig, with some human cells sprinkled in among the developing muscles and organs. But still, they did it. The researchers made the first human-pig chimeras, and they're hoping that the process can be improved to the point where pigs can be grown with specific human tissues or entire organs. There are lots of extra challenges involved in this kind of research, though, and it's controversial. For instance, nobody wants people growing pigs with, like, human brains. And we are a long way away from being able to do anything like that, but scientists researching chimeras are still being really careful about the kinds of experiments they try. And it's possible that someday this research will lead us to being able to grow functional, vital organs that people need.
Here with us today is Thompson, a success story surviving a pig chimera heart transplant. How do you feel getting a new pig heart?
Good. I feel normal again. I was not sure at first because I thought that perhaps it was tampering with our genetic makeup, and I would become a half pig. But I'm still human and kicking. It truly saved my life when finding an organ donor quickly.
Wow, that was really informative. Thank you for your time, Elliot Thompson. Now, in other news,
What's happening?
Cut.
Elliot, your organs are failing. We're going to have to perform a surgery on you. There are no human donors at this time, so we're going to have to use a pig chimera. There is no other choice. I'm sorry.
so Doc, what's happening?
Basically, what we're going to do is we're going to take pigs when there's still a couple of cells. Then we're going to edit the DNA and take out the portion for the organ that you need. Then we're going to take some of your DNA and insert it into the blastocyst to fill that gap. Then we will place this group of cells, this blastocyst, into a surrogate mother, which will then grow for 28 days. After that, we will perform a normal organ transplant and insert the pig's organ into yours, and hopefully there is no rejection.
Rejection?
Rejection is less of a probability now. You see, this is because the gestation rates and size of organs in pigs is similar to humans. But if the immune system of the pig is not compatible with yours, there is a slight chance of unintended consequences.
What are those consequences?
Right now we're not really sure. We haven't had many patients go through this surgery, as some are not willing to. We're going to have to go through many more surgeries and many more trials before we can be certain about any consequences or side effects from this transplant.
Thank you, Doc. Thanks to the human pig chimera transplant, my life has been saved. This technology could save many other lives like it. Oink oink. That's all, folks.
Alright. 3, 2, 1.